Hello and welcome to this teaching idea video on how to use a large model to teach how cells form tissues, how tissues form organs, and how organs go on to form organ systems. When running this activity, the two organs that I like to focus on, because it's nice and easy, are the small intestines and the pancreas. Right, historically this has been a little dull part of the specification to teach because students only really need to know that cells go to make tissues, tissues go to make organs, and organs go to make organ systems. However, it's a good opportunity to go and use a bit of modelling to get students to recall about the different functions of particular cells and how the different cells go and work together. Some key bits and bobs that you might want to consider before setting this up. Restrict the amount of cell types you use. I personally only use about 4 different types of cell type, as any more than that and the students start to become confused and can get them muddled up. Make sure that you've got plenty of cells cut out, because that helps reinforce that lots of cells go to make up tissues. And make sure that you've got a clear view in your head beforehand about roughly where you want the model to go. The following clip shows how this model can be used in practice. So, to start off the activity, go through what the different types of cells are with students. So go through, show them ciliated epithelial cells, ask them lots of questions about, right, so why are cells ciliated? What is it increasing, right? Hopefully they should come out that it's increasing surface area. Ask them why we need to increase the surface area, etc. Point out to them that in this case we're looking at ciliated cells in the small intestines. Why is there lots of mitochondria? What might they need that energy for? So they should come up with they need to actively transport stuff in. So you start off, go build yourself a circle of ciliated epithelial cells. Right, then ask students, right, so how is the food going to be forced all the way through the intestines? So hopefully they should come up with that it's going to be pushed along by muscles. So go and talk about muscular tissue. You then go and circle it with a layer of muscle cells, right? After that, hopefully you've already talked to them about the outside of all the organs is coated with epithelial tissue. So go and put a layer of epithelial cells around it. Get students to point out, right, so how are these 3 types of tissues different? So the ciliated cells are ciliated, so they've got greater surface area. The muscle cells, you've talked about, the muscle cells can contract and relax so they can move. The epithelial cells are nice and flat, so they're coated. After you've gone and talked about what's happening inside of the small intestines, go build yourself a small tube and lead to where the pancreas will be. Right, using the pancreas as an example is a really good idea because it enables you to talk about glandular tissue, right, without having to faff around and put more glandular tissue into what's already there. So go, put your glandular tissue in. Ask them, right, so what's the purpose of the glandular tissue? Relate it to the glandular tissue is going to have to produce enzymes that can then flow down into the small intestines. Then, as before, put a nice layer of epithelial cells around it just to encase it. Hopefully this video has given you some ideas how to teach this particular concept. If you've liked this video, please subscribe, like the video. There's links below to follow me on Facebook, Twitter, and there's a link below to my shop on test where you can go and get a full copy of this lesson. Please feel free to leave any comments or questions below and I will get back to you as quickly as possible. Thanks for watching and happy teaching.